Hello everyone, I am Mudasir Ahmad, postdoctoral research associate from Department of Food Science and Technology, University of Kashmir. I'm going to discuss about biopolymer based nanomaterials and delivery systems for encapsulation of bioactive compounds. Let's start with the background and the rationale of the studies. Biopolymers, particularly starch, they have several limitations in their native form, be it poor solubility, retrogradation, limited digestibility, or poor functional properties. There are several approaches that have been done in the past. They may be physical, chemical, or enzymatic modifications. And with these modifications, the applications in different food products have been increased. But now there's another technological approach that is size reduction of these biopolymers, and with it we expect improvement in techno-functional and nutraceutical properties as well, and they can be utilized as nano-enabled delivery vehicle for the carrier of bioactive compounds so that it can protect it against the digestive conditions. There are several methods for reduction of the size of the polymers. It may be physical methods, chemical methods, enzymatic methods, or combined methods. But we are particularly interested in ball milling, which works on the principle and uses frictional and impact with the high centrifugal forces, which causes the rapid particle size reduction. Apart from this, we have used the ultrasonication processes or mild chemical treatments like alkali hydrolysis. Let's first start with one of our study related to starch nanoparticle preparations by using ball milling. Here I'm going to represent only the outcome of the studies. The starch nanoparticles showed higher water absorption and less oil absorption capacity than the native starch. That means that we may require the less amount of the starch, and if it absorbs the less oil, it has an advantage in terms of the health that the if it absorbs the high oil is considered to be more disadvantageous for health. Further, starch nanoparticles prepared were amorphous, increased viscosity, and they had better thermal stability. And that means that we may require a less amount of the starch to attain the certain viscosity in particular products. In another study, we produced the starch nanoparticles by mild alkali hydrolysis and ultrasonication process. This slide describes how the starch nanoparticles were prepared. The starch 1.5% was initially hydrolyzed at 80 degree centigrade and then sonicated using ultrasonication, and then ethanol was added in the ratio of 1:2 in a drop-wise manner under continuous magnetic stir. The slurry was centrifuged and then lyophilized in a freeze dryer. What we achieved is that the particles had better thermal stability. There was increased in viscosity in the starch nanoparticles as compared to the native starch, and it had also good antioxidant properties. The starch nanoparticles were more amorphous and having increased water absorption and decreased oil absorption capacity. This study also confirms the similar reports what we have achieved in the previous studies. Then these nanoparticles, they can be used as a nano delivery vehicles for encapsulation purpose. Encapsulation is a technology in which we can trap the sensitive bioactive compounds in the internal structure of the biopolymers. There is a need to specifically protect the bioactive compounds which have higher bioactive properties because they are sensitive to the environment once they are extracted from the plant matrix. They are vulnerable to the environmental conditions, environmental stresses, or the harsh gastric conditions. We may need to achieve the targeted release where they can have the maximum absorption. There may be different types of the capsules. It may be in macro scale, it may be in micro scale, or in the nano scale. Encapsulation can be done by several methods, be it spray chilling, emulsifications, co-acerations, solvent evaporation, or extrusion. We have used electrospinning methods. You can have a look at. We are conducting few experiments. We use a spray dryer, freeze dryer, and supercritical fluids as well. The functions of the encapsulation in industry is very vast. Like it can stabilize the sensitive compounds during production, storage, and digestive conditions. It protects the compounds against the adverse heat, light, oxygen, and pH. It masks the unpleasant tastes and can improve the delivery of the bioactive compounds. It can increase in nanoform, particularly, it can increase the solubility and the permeability, which can help in better absorption and bioavailability of the bioactive compounds. In one of the study, we encapsulated catechin in starch nanoparticles. There is a need to encapsulate catechin because catechin is known to have high antioxidant activity, better than butylated hydroxytoluene or butylated hydroxyanisole, and it inhibits several carcinogenic processes. The major shortcomings for the catechin are low viability, less permeability, and degradation under digestive conditions. There are major sources for this catechin, be it green tea, berries, apple, grapes, and pear. But if we take it in as such, in the free from, we cannot have all the benefits from the catechin because it often degrades in the digestive tract. This slide shows how we prepared the starch catechin nanocapsules, and the starch nanocapsules were then characterized by several techniques. We particularly measured the release behavior under simulated gastric conditions, and few tests like anti-diabetic and anti-obesity tests were also conducted. They were conducted by measuring the percentage of enzyme inhibitions like alpha glycosidase, cholesterol esterase, or pancreatic lipase. What we achieve is that the nanocapsules of the catechin were more beneficial than free catechin. That means that it had actually the higher bioactivity than the free catechin under digestive conditions. And it's because that there was slow and controlled release from the capsules in the gastrointestinal conditions. And because of that, the bioactivity was better retained after digestional conditions. In another study, we encapsulated folic acid and saffron anthocyanins. Folic acid is of paramount importance from the health perspective. It is susceptible to degradation under acidic conditions, and unmetabolized folic acid in blood is known to develop certain cancers and cardiovascular diseases, anemia, and other adverse health effects. But upon encapsulation, there was the maximum amount of the folic acid in the intestinal section where it can be absorbed. So the percentage of the unmetabolized folic acid can be decreased upon encapsulation. Saffron anthocyanins, they have tremendous bioactive properties, but it is difficult to transfer anthocyanin to the intestinal tract because it is highly unstable in the adverse gastric conditions and environmental conditions like light and heat. Encapsulation brings the stabilization of the saffron anthocyanins and increases their bioavailability. In another study, we compared this micro and the nano encapsulation. We encapsulated probiotics, Pediococcus acidilacticine. This study was actually carried out to know whether the nanoparticles can be suitable for the encapsulation of probiotics. What we achieve is that the nanoparticles could not hold the probiotics inside the cells well, because during gastrointestinal release, the cells were abruptly released just after the 5 minutes, and there was no survivability, no cell content was found after 30 and the 60 minutes. While as in microparticles, it was slowly released. The cells were slowly released in the intestinal section. The similar case was found in thermal resistance. Cells could not hold a higher temperature in nano-encapsulation form, where micro-encapsulated probiotics showed some thermal resistance at higher temperatures. And then what we conclude is that nano-starch particles could not offer thermal protection to probiotic cells, and nano-encapsulated probiotic cells display lower viability compared to cells in native starch. And finally, we concluded that nano-sized polymers may not be stable for encapsulation of probiotics. In another study, we encapsulated resveratrol in starch nanoparticles. Resveratrol is highly anti-carcinogenic, anti-inflammatory, anti-obesity, and anti-diabetic. But it is unstable to light, heat, and enzymes, and less soluble and less stable in gastrointestinal conditions. And because of this, we encapsulated resveratrol in starch nanoparticles, and similar tests were conducted. What we observe is that the encapsulation efficiency was found to be around 50 to 60%. And maximum amount was released in the intestinal section. The study was confirmed. The presence of the resveratrol in the capsules was confirmed by SEM and FTR spectroscopy. The capsules were more amorphous in character. There was decrease in the crystallinity of the starch after formation of nanocapsules. The bioactive properties of the free and nano-encapsulated resveratrol was also conducted, and what we found is that the encapsulated resveratrol offers better protection to its bioactivity than free resveratrol. And finally, it was concluded that the resveratrol was released in higher content in intestinal section, and antidiabetic and anti-obesity activity was better found in encapsulated resveratrol than in non-encapsulated resveratrol. Now, applications in the food industry. These nanocapsules, that are nano-encapsulated bioactive compounds, can be used in food fortification, like we developed novel functional snacks. And then the snacks were characterized to measure the resveratrol content after processing. The molecular characterization, physical properties of the product was also analyzed. The antidiabetic, anti-obesity, antioxidant activity were also analyzed to know the effect of encapsulated resveratrol, and it was compared with the resveratrol without encapsulation. The sensory properties were also conducted to know if there was change in the consumer acceptability of the product. And what we found is that the encapsulated resveratrol snacks showed better anti-diabetic and antioxidant and anti-diabetic activity than snacks with free resveratrol. And there was no differences in the sensory score of the snacks. It was much comparable to the native product. And what we conclude is that encapsulation process can prevent the thermal degradation of bioactives during processing. And this study highlights the nutraceutical importance of using bioactive compounds in an encapsulated form in various food formulations. This slide shows the future scope of the study. The research should be carried out to check the bioavailability and cell permeability of nanoparticles to validate the exact fit of nanoencapsulation of bioactive compounds. The encapsulated bioactive compounds can be used in different food formulations. And we may need to check the bioavailability and bioactivity of encapsulated compounds under in vivo and in vitro conditions. And this slide shows some of my key publications with impact on more than 4. These are my awards and achievements gained in a short period of my research career. This is a letter of appreciation from Honorable Vice Chancellor for receiving Young Investigator Award in one of the international conference. Thank you very much. I hope you have enjoyed.